Okay. Hello, everyone. I'm going to present our work consisting of the integration of clinical and genetic data for precision medicine to be applied in oncology projects. This work was carried out by medical doctors, biology scientists, and bioinformatics, and the authors are listed below. My name is Lucas Mulan. I'm an advanced student of biotechnology and bioinformatics, and I have been working on the development of DNA and RNA analysis for application in oncology medicine for the treatment of tumors and cancers in what is known as precision medicine. So to begin, precision medicine, as defined by the US National Library of Medicine, is an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. So, to this end, the general aim is to work towards a more personalized and customized way of treating certain types of cancers. For this purpose, a research project for precision medicine in oncology was developed. Said project is titled Tumor Genomic Actionability Map of Argentina, and it is known as Magenta for its acronym in Spanish. This project is carried out through a public and private associative consortium consisting of the Italian Hospital of Buenos Aires, the Multidisciplinary Institute of Cell Biology, and the Institute of Translational Medicine and Biomedical Engineering. The listed acronyms are given by their names in Spanish. So the Magenta project consists of the studying of clinical information of hospital patients combined with the genomic information derived from 52 tumor-related genes, with the objective of characterizing the Argentine's population genomic profile. The Magenta project aims to classify tumors from a therapeutic point of view in order to provide a new paradigm in diagnostic, treatment, and follow-up of each patient. The integration of clinical and genomic information involves combining data that resides in different sources and providing users with a unified view of it. This integration, if carried out manually, is time-consuming and requires technical knowledge. At the same time, the manual procedure might produce transcription errors, and as more samples are included, the repetitive task stagnates the development and consumes time which could be spent elsewhere. So due to the aforementioned problem, the objective of this work is derived, which is to implement a programmatic strategy to automate the integration of clinical and genomic information of the Magenta project to facilitate and reduce operator time and the probability of data transcription errors. For context, this work was carried out in the Italian Hospital of Buenos Aires, a highly complex hospital that has had an electronic medical record for more than 20 years and is certified at level 7 by the Healthcare Information and Management Systems Society. It is also certified by the Joint Commission and by the College of American Pathologists. For the development of this first implementation, 186 patients were included who had had a tissue sample extracted from a previous surgery or biopsy. These patients were selected by medics from the oncology and pathological anatomy departments and consented to take part in the development of this project and were otherwise excluded to participate. In order to provide a solution for the integration of clinical and genomic information, we elaborated this layered system in a process known as ETL, which means extract, transform, and load. The information is first extracted from different genomic sequencers and from the electronic health record containing clinical information of patients. then uploaded to a form, transformed and processed by computer scripts, and finally loaded into an analysis and visualization platform. So let's start with the origin of information layer. First, clinical data from the electronic health record included information such as age, gender, histories of diabetes, smoking, and alcoholism, and information regarding their tumors, such as the type of cancer, the tumor stage, and whether they had a surgery from the tumor source. Alongside the clinical information, the first genomic information gathered comes from the incorporation of next-generation sequencing technologies, which was used with an oncomine focus assay panel, which analyzes 52 clinically relevant solid tumor genes, which includes 35 single nucleotide variation and insertion deletion genes, and 23 fusion genes. These genomic sequencers are managed and interpreted by an ion reporter software and will later be used to access the sequence information. Moreover, in another laboratory, the mitochondrial ancestry of the patients was analyzed, which makes it possible to determine the haplogroups and the maternal ancestry. Once this information is obtained, it has to be unified in the storage layer. For this, we use a tool named RedCap, which is a data capture software and allowed the creation of dynamic forms in order to upload the information provided by the previous layer, which includes clinical data and genomic metadata. For this purpose, RedCap version 7.0.11 was used. After this, the processing layer comes into action to formally integrate these sources of information loaded onto the RedCap forms. To achieve this, we developed a bioinformatic pipeline that processes the uploaded clinical and genomic information and transforms it so it can be uploaded into an analysis and visualization platform using application programming interfaces provided by RedCap, Ion Reporter, and CBioPortal, which permitted programmatic access to these platforms. And finally, the visualization layer is done through CBioPortal for Cancer Genomics, which is a web platform that provides statistical analysis and visualization tools of the uploaded information. This platform requires a defined file system structure, which the developed pipeline produces, and it allows high-quality analysis for further use in the Magenta project. Both the CBioPortal and RedCap platform were implemented on the local servers of the Italian hospital. In this picture, we can see some of the many databases that CBioPortal integrates and converges them into one unified platform that provides detailed analysis on many aspects of the biology of the cancers studied. So entering now in the results section, for the RedCap capture of data, 8 loading forms were implemented, which were managed by each department specialized in that area, which includes the pathological anatomy and oncology departments and the sequence laboratories. So here we can see an overview on how these forms result in the RedCap platform, where each green dot means that the patient has loaded information on that column's topic. After the upload, the developed integration tool processes and annotates the information uploaded to RedCap through Python and R language scripts, which finally produce information in CBioPortal accepted format for export. This is an overview scheme of the scripts involved. It starts by downloading data from the RedCap forms, filtering it, and then annotating it, enriching its content, while also creating log files that serve as a review of the process. Afterwards, it captures data from the Ion Reporter system, and then it separates the information in several files that contain specific information as it is requested by the format file system of CBioPortal. Finally, it exports them to that visualization and analysis platform. These scripts were then run through the Cromwell Pipeline Manager, which runs some scripts in series and others in parallel according to specifications. For this initial run, it took around 30 minutes to process information of the 186 patients. And we can see that the process which took the longest was the downloading of the sequence files and reports. In this picture, we can see the first output of the results that the CBioPortal platform provides. All these graphics and tables can be further filtered by the graphical user interface provided by the platform. Here is another capture where we can see more statistical tools provided by CBioPortal. All these results can be accessed at the website shown at the bottom right corner of the screen. Here we show a map of the instances where CBioPortal is used around the world. We can see that the Magenta project is the only one in Argentina and one of the 3 in South America. Now, to conclude, the developed pipeline allowed a successful integration tool from different sources of information containing clinical and genomic data. This integration proved to be an enriching tool to further develop precision medicine projects in oncology, in which information must be updated constantly at a specific frequency. Moreover, it proved to decrease manual work time for integrating clinical and genomic information, therefore making time available to expand on analytic areas of the project. It also allowed anyone to carry out the integration process without the need of having technical knowledge that CBioPortal requires for this operation. Future versions and enriching tools will expand the reaches of the Magenta project. Finally, here are some of the names and faces involved in this project and the continuation of it, along with their workspaces listed by their official Spanish names. Thank you very much for your attention. Muchas gracias, and I hope you enjoyed it.